Hello everyone, I am Fatima Sultana. I work as a research aide in Dr. Reddy's lab at Texas Tech Health Science Center. Our lab has been dedicated to studying Alzheimer's disease, dementia, and aging for many years, contributing significantly to the field through numerous publications. Today, I am excited to present one of our recent review paper focused on unraveling the complexities of human brain, shedding light on structural and functional changes that occur in neurodegenerative disease. Let's begin with background information. The brain is one of the most complex and vital organ in the human body. It consists of three primary components such as cerebrum, cerebellum, and brainstem. The mature brain consists of 100 billion of specialized cells known as neurons, each dedicated to processing vast amount of information. It is also responsible for controlling thought, emotion, memory, and bodily function. It is the part of the central nervous system along with the spinal cord, and it is protected by the skull, cerebrospinal fluid, and meninges. So in this slide, I'm going to talk about the major parts of the brain. The brain can be broadly divided into the three major parts based on the structure and function. The first one is forebrain or prosencephalon. The forebrain is the largest and most complex part of the brain, responsible for higher cognitive function, sensory processing, and voluntary action. It is further divided into two main regions. First one is cerebrum, i- which include two hemispheres, left and right, connected by the corpus callosum, each with four lobes: frontal lobes, parietal lobes, temporal lobe, and occipital lobe. On the picture, we can see the frontal lobe, parietal lobe, and occipital lobe, and temporal lobe. And the function is thought, reasoning, and decision making. Sensory perception, for example, touch, vision, hearing, voluntary motor control, and language and communication. As I said in my previous slide that forebrain is divided into two major parts. First one is cerebrum that I already talked about it. The next one is diencephalon. So diencephalon contains thalamus, hypothalamus, and epithalamus. Thalamus relays sensory and motor signals to the cerebral cortex. Hypothalamus maintains hemostasis and regulates the endocrine system, and epithalamus, which contains pineal gland as well, which is actually regulate our sleep cycle. The overall function of diencephalon are integrate and relay sensory information and regulate emotion, temperature, hunger, and thirst. On the picture, we can see the structure of diencephalon, thalamus, hypothalamus, and, and other parts of the brain. So the next part of the brain is midbrain or mesencephalon. Midbrain is the smallest part of the brain located between the forebrain and hindbrain. The key structure of the midbrain are tectum or tegmentum, cerebrum peduncles, and substantia nigra. The function of tectum and tegmentum is control reflexes related to vision and hearing, such as, for example, turning your head towards a loud sound. Then cerebral peduncle. Cerebral peduncle function is relay motor signals from the cerebral cortex to the other part of the brain. And finally, substantial nigra, which is responsible for movement and reward. On the picture, we can see the midbrain structure, the forebrain, midbrain, and hindbrain, and other part of the brain as well. So the next part is hindbrain and the rhombencephalon. The hindbrain is located at the base of the brain and essential for basic life function and coordination. It includes cerebellum, pons, and medulla oblongata. Cerebellum is responsible for voluntary movement, posture, and balance, and plays a role in motor learning and fine tuning movement. The next important part is pons, which act as a bridge between the cerebrum and cerebellum, regulate the breathing, sleep, and arousal. And then medulla oblongata. Medulla oblongata mainly control our autonomic functions such as heart rate, blood pressure, respiration, and it also facilitates the reflex action like swallowing, sneezing, and coughing. The other important part of the brain are spinal cord, which is essential part of the central nervous system, and the spinal cord runs from the brainstem to the lumbar area and it ends at lumbar 1 to lumbar 2 in adults. Terminating at the conus medullaris, the spinal cord gives way to the cauda equina veil, a bundle of nerve roots within the vertebral canal. The spinal cord facilitates the exchange of information in both directions between the brain and the peripheral nervous system. The spinal cord is structurally composed of both gray matter and white matter. The three primary functions of spinal cord in the nervous system include synchronizing reflexes, sending sensory information from the body to the other part of the brain, and transferring motor commands from the brain to the body. So on the picture we can see the conus medullaris and cauda equina and vertebral cord. So those are the main major parts of the brain that I already talked about. As we know, this brain is complex organ and it has lots of structure on it. So, but we talked only the major parts of the brain. So now I'm going to talk about the synapses of the brain. A synapse is the junction where neurons communicate with each other with other cells. For example, muscles and brain. There are two types of synapses. One is chemical synapses, another is electric synapses. Chemical synapses mainly they are neurotransmitters which are used to transmit signal across a synaptic cleft, a gap between the neurons. For example, serotonin, dopamine, and glutamate. In case of electric synapses, they use gap junction for direct transmission of electric signals, allow faster communication, but are less common in the brain. So on the picture, we can see the synapses of the brain. One is chemical synapses, another is electric synapses. So on that slide, I'm going to talking about the blood-brain barrier. The blood-brain barrier is a highly selective permeability barrier that separates the circulating blood from the brain and the extracellular fluid in the central nervous system. It is a crucial component in maintaining the brain stable environment, maintaining the brain complex and dynamic environment, and protecting it from potentially harmful substances while allowing the essential nutrient to pass through. The blood-brain barrier is primarily composed of endothelial cells that line the brain capillaries. Those cells are highly packed together, forming tight junctions that restrict the passage of substances. Those junctions between the endothelial cells are formed by the proteins such as chlorine and chlorines. They prevent the passage of most molecules between the cells, ensuring the substance must pass through the cell rather than between them. So on the picture we can see the a healthy blood vein variant and other component. So now I'm going to talk about the electrophysiology of the brain. This field focuses on how neurons generate and transmit electrical signal, which are essential for brain function and communication. Neural gradient maintain a voltage gradient across their cell membrane known as the resting membrane potential, which is typically around minus 70 millivolt. This gradient is created by the distribution of ions, primarily sodium, potassium, chloride, calcium across the membrane. When a neuron's membrane potential becomes significantly depolarized, an action potential is generated. An action potential is a rapid rise and the fall in the membrane potential of a specific location, causing adjacent areas to depolarize similarly. This occurs in excitable cells, including neurons, muscle cell, endocrine cell, glomerulus cell, and some plant cells. So on the picture, we can see the action potential in a neuron highlighting the phases of depolarization and repolarization. During depolarization, sodium ion influx and making the inside of the neuron more positive. This is followed by repolarization where potassium ion is flux restoring the negative membrane potential. So now I am going to talk about the disparities in brain structure in between male and female. So on the diagram we can observe the variation in brain structure between the male and females. Some regions appear enlarged in male compared to female, where, while conversely certain structure exhibit larger proportion in female than in male. So on the picture we can see the male has greater amount of total volume of the brain, white matter, CSF, total corpus callosum, cerebellum, pons, etc. So on the other hand, we can see the female has greater amount of gray matter, hippocampus, frontoorbital cortex, superior frontal, and lingual gyri. So in this slide, I'm going to talk about the brain structural and functional changes in the neurodegenerative disease. So now I'm going to talk about Alzheimer disease and mild cognitive impaired, which is MCI. So Alzheimer disease is a progressive neurodegenerative disorder characterized by significant structural and functional changes in the brain. So what are the changes occur in Alzheimer disease? First one, Alzheimer disease causes damage to and multifunction in the brain neurons, which can lead to a breakdown in the connection between the neural network and the shrinkage of the certain brain regions. Intercellular aggregates of hyper phosphorylated tau protein interfere with the structural integrity and transport system of neurons, leading to cell death and as well as a decrease in brain volume. Alzheimer's disease initially affects the endoreal cortex and hippocampus, which is critical for memory, before progressing to the neocortex, impacting the logic, language, and social interaction, and eventually causes widespread brain damage. During this time, there is also loss of brain tissue, which lead to an expansion of brain's ventricle and leading to ventricular enlargement. So now I'm going to talk about the functional changes occur in Alzheimer's and MCI. The first of all is first of all, short-term memory is affected early due to the hippocampal dysfunction. Difficulty with planning, problem solving, and decision making arises as a frontal lobe connection deteriorate. Language and spatial issue. Progression into the temporal and parietal lobes lead to language deficit and impaired spatial navigation. Reduced glucose metabolism in the posterior carniculate cortex, parietal lobe, and temporal region. There is also impairment in connectivity, which is essential for resting state brain function. So those are the picture of MRI. MRI images compared to control healthy people, we can see the Alzheimer's and MCA patient brain has shrinkage and atrophy. So now I'm going to talk about what are the structural changes occur in Parkinson disease or PD. Parkinson disease is characterized by the degeneration of dopaminergic neuron in the substantial nigra, accumulation of Lewy bodies, and widespread structural and functional changes in the basal ganglia, cortex, and other brain regions. Those changes disrupt the motor circuit, cognitive function, and autonomic processes, leading to both motor and non-motor symptoms. Significant loss of dopaminergic neuron leading to reduced dopamine level in the stratum, which impaired motor control and contributes to hallmark symptoms like tremor, rigidity, and bradykinesia. There is also intracellular aggregates of alpha synuclein proteins found in the substantial nigra, other brainstem area, and cortical region, which disrupt the neural function and contribute the neurodegeneration. So on the picture, we can see the pathology of Parkinson diseases. We can see the, the other factor which contribute these Parkinson diseases. Patients with PD exhibit atrophy in several cortical and subcortical brain regions, which lead to the brain decreasing volume. Remarkably, it is observed in the PD patient had a larger volume in the temporaloparietal junction, parietal lobe, frontal lobe, anterior carniculate cortex, basal ganglia, thalamus, and insula. PD affects the prefrontal cortex, which is crucial for cognitive function. Now I'm going to talk about what are the functional changes occurred in PD. In PD, we can see that there is impairment of motor planning and execution. Our activity in the indirect pathway of the basal ganglia increases motor inhibition. Decreased activity in the direct pathway reduce movement facilitation. Functional changes in hypothalamic-cortical circuit affect the voluntary movement. Dysfunction in the prefrontal cortex and associated network lead to executive dysfunction, impaired decision making, and attention deficit. So next, movement in Huntington disease. Now I am going to talk about how do the structural changes occur in Huntington disease. Huntington disease is a progressive neurodegenerative disorder caused by a CAG trinucleated repeat extension in the HTTG, leading to widespread structural and functional brain changes. Striatal atrophy, chordate nucleus, and putamen. Significant volume loss and neural degeneration in the early stages. There is also cortical atrophy in the frontal and temporal lobes, which leading to progressive thinning, particularly in the region associated with cognitive and behavioral behavior. White matter degeneration reduces connectivity between the striatum and the cortex due to degeneration of the white matter tract. So on the picture, we can see the pathology of Huntington disease. So at the same time, we can see the healthy neurons and the damaged neuron and the which chromosome and gene is responsible for Huntington disease. Now move on functional changes. Functional changes such as motor symptoms such as chorea, which is involuntary movement, executive dysfunction such as impaired planning, decision making, memory deficit, and difficulties with attention and learning. Emotional and behavioral changes such as irritability, apathy, and depression. Functional connectivity is altered in brain network involved in movement, cognition, and emotion. So that's all for today. I hope you like my videos. Thank you for your attention. Now I'm going to talk about the summary of my whole presentation. The intricate interplay between the brain structure and function emphasizes its adaptability across the lifespan, and the structural and functional changes in neurodegenerative diseases like Alzheimer's, Parkinson, and Huntington disease. Distant pathological hallmarks such as amyloid plaque, dopaminergic neuron loss, and Huntington aggregates lead to progressive neural dysfunction and symptoms. Embracing the diversity, interdisciplinary approaches, and personal medicine is neuroscience research can improve diagnostic, treatment, and patient outcome while advancing our understanding of the brain complexity. Thank you all. I hope everybody enjoy my video. So thank you all for your attention. So if you wanted to read more about brain structure and function in healthy and disease states, we can look this paper which published in Aging Research Review. And if you have also any question, please contact us reddylab.research@gmail.com or reddylab@t2acc.edu.